welcome grade 10s. Welcome to yet another awesome, exciting lesson of life sciences. I hope that you have your textbook with you or any kind of resource that you're using, if you've got a tablet or whatever. And please make sure that you have your notes from last week when we did plant tissue. Make sure that you have the plant tissue notes handy as well, because I'm going to be asking you questions along the way, and we're going to refer to our plant tissue notes from last week very often. Okay, welcome again. Today we are going to look at the plant organs, and I know whenever I just say the word plant, I know my students at school always go, ah, all right. No, come on, be excited. Plants are wonderful, plants are beautiful, right? And plants are one of the most important things in our biosphere. So we have to look after them, and if we have to study them, we can understand them better. Okay, today we're going to look at, we're starting a whole series on plant organs. Now, if you can remember last week, you would have dealt with a whole section, and I know sometimes they looked quite difficult and they all look the same, but you dealt with plant tissues. Okay, so when we're looking at plant tissues, why were we looking at plant tissues? I want you to have a look at the picture that I've got on the board in front of you here. Right, now if I twirl down to the bottom, I want you to start over here. Where are we going? So if I start over here, if you can remember at the very start, oopsie, let's get the pen going. If we started at the very beginning, we started with this whole concept of a plant cell. And before we even looked at the plant cell, what did we start? We looked at, oh, I'm trying to get my colors ready today. We looked at the whole biochemistry, okay? And if you look at how we're progressing, where have we started? We started looking at a plant cell, but before, or a cell, and before we looked at it, we looked at how things were made, okay? Now, last week, you looked at this whole concept of tissue. What is a tissue? We looked at a very simple, basic, right, concept called a cell. Okay. Now, a cell, when we look at the cell was just a unit. It was a model of something that we looked at. And when you started looking at tissue last week, we noticed that cells, all right, when you looked at phloem, you looked at xylem, right, you looked at parenchyma, you looked at sclerenchyma, you looked at epidermal tissue, and what did you notice? That plant cells look different because they have different functions, all right? And that was the term you learned last week. When we take a group of cells, right, here's a group, okay, you're going to be doing mammalian tissue soon. Here's a group, specifically the one we're going to look at today, and you put all the cells that look similar together and they perform a similar function, all right, we got this whole concept of a tissue. Now this week we are going to take the tissues that you looked at, all of the phloem, the xylem, the epidermis, the parenchyma, the sclerenchyma, and we are now going to put it into this structure that we call an organ. Okay, an organ is a group of tissues, right? We've got a group of tissues, we've got different kinds of tissues, and all depending on what my organ needs to do, I'm going to structure or put these tissues into their different places. Okay, and today we're going to concentrate specifically on the leaf. Okay. So now, I hope you've got everything together. I'm going to give you, all right, just 1 minute. Quickly, I'm going to, throughout this, I'm going to give you just little small questions, just to try and see if you can you need to start thinking about, okay, this is what my plant tissue looked like and what it did. This is what my organ is. How am I going to relate the functions and the structure of my plant tissue to what I need in the organ? Okay, so I'm going to give you 1 minute. You don't have to write too much down. The 2 questions that I want you to answer. What is the main function of a leaf? Okay, now, thinking back, all right, I've given you a little hint. Think back to the plant tissues that you have done. What tissues am I going to find in the leaf that will be able to assist the leaf in performing its function? Okay, I'm going to give you 1 minute. Okay, 1 minute, and then we're coming back to see if you've got the concept. Your 1 minute starts now. Welcome back. I'm sure you were able to answer the questions. The first one, I hope you were able to, and let's see how you did with the second one. Okay, the question was, what is the main function of a leaf? And I hope you had this as your answer. Photosynthesis. All right. Now, photosynthesis, all right, is how the plant is able to take sunlight and make food. Now, in the beginning of the cell, you looked at a special organelle called a chloroplast. Right, and we know that that is where photosynthesis is going to occur. So let's see how you managed. Could you remember from last week, what tissues am I going to find in the leaf that will be able to assist the leaf in performing its function? Okay, one of the ones you should have had, all right, was, can you remember last week we did a tissue called parenchyma. Now, parenchyma, when it's filled with chloroplasts, is called chlorenchyma. And if the chlorenchyma is filled with chloroplasts, that means that it is going to help with photosynthesis. Okay, now, I wonder how many of you thought of a few of the other tissues. For example, did you think of xylem that brings water up to the leaf for photosynthesis? Did you maybe think of phloem that takes the food that we make from photosynthesis and passes it down again? All right, and maybe did you think of some kind of strengthening tissue, parenchyma, right, that is going to help support the leaf so that the leaf can always face the sun. Right, so how did you guys do? Of all of these, I'm hoping that you were able, right, to definitely, the other ones might not have been as easy, but I hope you definitely got the parenchyma one. Okay, so let us start with the leaf. I'm going to give you some time again. What you're going to look at, we're going to look at what a leaf looks on the outside and what a leaf looks on the inside. So the first thing I'm going to do is to ask you, and I'm going to give you 1 minute, right? Just a short little time. Your book's out, and I want to give you just a short little bit of time, as I just said. I want you to label what the external, the outside of a leaf looks like. Okay, your time starts now. Welcome back again. I hope you were able to label the diagrams of a simple leaf. Okay, shall we go over the labels and see how many of you got right? Okay, guys, so when we look at the leaf, right, we know that the bottom layer over here you should have had is a stem. Because what a leaf is? A leaf is going to be on a stem, and we're going to attach the leaf to this little small stalk over here called a petiole. All right, and this petiole needs to be able to hold the leaf so that the leaf can face the sunlight. Okay, so the leaf needs to be able to face the sunlight. Now we look at the structure that you guys know as a leaf. Okay, let's start on a few things. The first thing, you will notice that a leaf usually has a top part. And the top of some part of something we call the apex. A leaf also has an outline, right? As you can see, it has a definite ending, and we call that the margin. Now, whenever you guys draw a leaf, I know for a fact this is usually the leaf that you draw, because you draw this main line down the middle, right? And that is called a midrib. And a midrib, as it says exactly, is the middle of the leaf. And that midrib, right, and the little tributaries or little parts that going over here are actually the veins. So when I say veins, all right, the same as you and us, except there doesn't have a blood system, you need to be thinking, all right, transport. Okay, transport. Now, the leaf itself, what you usually refer to as a leaf blade. So when you look at the leaf, it's the actual leaf that you see. Is not called a leaf blade. Well, it is, but technically speaking, we want the biological term, and that is called the lamina. Okay, so pretty simple. The word's not too intense, and that is the external structure of the leaf. Now, all right, when we look at leaves, and this is something that we're going to continue on next week, is this whole concept of a dicotyledonous plant and a monocotyledonous plant, because we're going to see that there are slight differences when we look at the plant. Just by looking at certain aspects, we can tell if it's dicot or if it's monocot. Okay, so I've said here, what does this mean? What does it mean when I say it's a monocot or a dicot? If you have the chance, I want you to get a milli, all right, a milli seed, all right, or like same as like you have popcorn, and I want you to get a bean seed, all right? And when you take a milli and you take a bean, I want you to cut it open, all right? Sometimes it's a bit easier if you soak it in a little bit of water for a while, okay? Then I want you to take a sharp knife and I want you to cut it open. Okay, now why did I say that? Because when I talk about a monocotyledon or a dicotyledon, mono means one, okay? And dicotyledon means two. Now, what on earth does that mean? Let me show you this diagram over here. When I break the seed open, when I take my milli and I break it open, I find that it only has one piece inside. And that little piece we call a cotyledon, okay? But if I take my bean seed and I cut it open, I find that it separates and falls into two equal parts. That means that it is dicot. Dicot means two, as you can see here, and monocot means one, a single one. Now, because of that monocotyledon and because of that dicotyledon, what does that mean? We're going to see the leaf, all right, and we're going to do it next week, so I'm not going to go into too much detail. We can see the leaves are slightly different, the roots look slightly different, all right, and the flowers look slightly different. So what we're going to concentrate on today, we're going to concentrate on a dicotyledonous leaf. Okay, now when we look at the leaf, we're going to look at what it looks like on the inside. But before we do that, I'm going to give you a quick break, right? We're going to come back just now. Please make sure that your textbook and your notes and everything, right, is turned to the leaf so that you can follow with me when we go through it. Have a quick break and we'll see you now. Well, guys, I hope you had a nice little stretch and that your notes and everything that your teacher's given or your textbook is open. Remember, you need to follow me while I am helping you with the lesson. Okay, now what I'm going to put up on the board now is a diagram. Now, it's a very simple diagram. What I'm starting off with is for you to be able to handle it quickly. It sounds funny when I say that. I'm going to give you a very simple diagram of what the leaf looks like on the inside. And when we do that, we're going to look at three main parts. So when I look at the leaf, I'm going to concentrate on there's three main parts that we're going to concentrate on. The outer parts, the epidermis. Then we're going to see that there's an inner part, and then we're going to see there's a specialized part called the veins. Okay? So I hope you have your textbooks out, so now I'm going to go through it with you. I want you to have a look at the diagram in front of you here. Okay. This is what we call a dorsiventral leaf. Okay, what does dorsiventral mean? Dorsi means top. Over here, I have a top of a leaf, and over here, I have a bottom of the leaf. When I look at the leaf, if I look at this diagram, I want you to quickly look at me to see what picture you are looking at. Okay, it's called a longitudinal section. A longitudinal section means I've taken the leaf, all right, and I've cut it from top to bottom. I've cut it from top to bottom. And then I look at it, and what I'm looking at is all the cells that I can see from the top to the bottom. Okay? A longitudinal section, a long, top to bottom. Now, if we go back to the diagram over here, we notice, right? We will notice that this is the top and that is the bottom. Okay, and when we look at the top and the bottom, we notice that we can tell differences. But the first thing we're going to look at is this pink area over here. Now I'm going to use a different color so that you can see, because it's pink here. Do you notice this area that I've outlined in blue? Now I want you to look at your tissues. When you looked at your tissues, all right, you looked at a tissue called an epidermal tissue. All right, and I think I even did the plant tissues with me. When we did the plant tissues, we looked at a single cell epidermal tissue, okay? And we see that we find an epidermal tissue at the top and at the bottom. Now, if you notice, adaxial means top, and an abaxial means the bottom. So we're going to find epidermal tissue. Go back to your epidermal notes. Go back to your plant tissue notes and have a look. All right, my epidermal tissue looked a lot like that. Right, very simple cells. So I'm going to find that lining the top and the bottom. What do you think the function is there for? Epidermis, the same as my epidermis of the skin, protects. Now, I'm going to give you one quick little job to look at. I need you to think. Look at the question. Draw and label a diagram of a specialized epidermal cell found on the lower surface of the leaf. Let me go back here. When I look at my diagram, on the bottom of my leaf is a specialized epidermal cell because, all right, something needs to happen at the bottom that doesn't happen at the top. I'm going to give you only 1 minute. It doesn't need to be a masterpiece. All I want you to draw is those specialized cells that are able to, here's the hint, open and close, that are found at the bottom. 1 minute. It's a quick diagram. Your time starts now. Okay, hope you got the diagram. Guys, you need to understand that your plant tissue section is very much a part of your plant organ. Remember what we said in the beginning, tissues make up organs. Right, so what was I looking for? I gave you that hint, it opens and closes. I hope you drew for me the stomata. All right, I'm hoping that you drew the stomata and you had a word here, you had guard cell. Okay, and you had nucleus. And you had chloroplasts. Chloroplasts. All right, what else should you have had? Oopsie daisy. Here, what do we know about the stomata? The inner walls are thickened. So thick inner walls. See guys, it doesn't have to be a masterpiece. It just has to be a little diagram that I understand. All right, and a thin outer wall. Thin outer wall. And don't forget the hole in the middle, which is called the stoma. Okay, so remember when we go back to our tissues, all right, it's essential that your tissues, you're going to link your tissues with your plant organs. So have the notes there together. Have your plant tissue, have a nice big A3 shaped paper where you've got pictures of all the plant tissues, what they do, where they are, and here you've got a diagram of the leaf, all right? And you can then even use coloring in pencils. You can color code the different tissues, right, to the different parts of the leaf. All right, so I said to you that a leaf is made up of 3 parts. Okay, now the next one might be a word that you're not familiar with. This word is called the mesophyll. And if I were to make use of the analogy of a sandwich, right, we had the epidermis at the top, the epidermis at the bottom. This is the filling of my sandwich. Meso means middle. All right, meso means middle. And obviously, this makes up the majority of the tissue in the leaf. Also, using the colors that we have used, you should also have some idea that it's this tissue, all right, that is going to be responsible for photosynthesis. Okay, guys, so what have we got? Underneath the epidermis, all right, I have these long, elongated cells. They look like this. Why do I elongate them? Elongated means increase surface area. All right, so I've got lots and lots of space. Now, why do I want to have lots of them? Because look what I'm going to have coming down. Sunlight. Sunlight comes down. It must go through the epidermis and it must hit my palisade tissue. Because of what have I got in my palisade tissue? Wow, I'm packed full with chloroplasts. All right, and what do I need for photosynthesis to occur? Chloroplasts. So straight under the surface of my leaf, here are all these long cells. The sunlight comes down, and you're going to do photosynthesis next year, so you're going to have to remember this next year. And the photosynthesis comes through the epidermis, and it hits all these cells at the top that are nice and big and long, and they're filled with chloroplasts. Because what is the function of my leaf? Photosynthesis. So I must be able, my leaf must be able, all right, to perform its function, which is photosynthesis, to the best of its ability. Okay, so if you have a look at the leaf, what do I see? I see it's packed full of this tissue, all right, called we're going to call mesophyll, all right, palisade mesophyll, the top one over here. All of this, let me change back to blue, then it's easier. Okay, but if you notice, the palisade is on the top. It's not on the bottom. Why is it not at the bottom? Because where does sunlight trigger? Sunlight comes from the top. Sunlight doesn't come from the bottom. So I want my cells that are going to, all right, be responsible for photosynthesis to also be, all right, at the top. Then I have, now that's your palisade mesophyll. Then I have spongy mesophyll. Now spongy are round, okay? They do not have as many chloroplasts as the palisade, but they do have chloroplasts that can photosynthesize. But look at their shape. In their being round, okay, look what they can do. They create these air cavities. Guys, you might not have done photosynthesis yet, but photosynthesis needs carbon dioxide. All right, photosynthesis needs carbon dioxide. And how do I get the carbon dioxide inside? I make sure that my stomata are open, and what comes in? Air. And what is in the air? Carbon dioxide. Look at all these spaces. What can I fill with all these spaces? I can fill it with air. And what's in the air? Carbon dioxide. And what do I need for photosynthesis? I need carbon dioxide for photosynthesis. So look at the structure of the leaf. It's got cells in that have got full of chloroplasts to photosynthesize, and also it's got cells whose round shapes enable lots of air chambers and air spaces, all right, to exist, so that I can keep this air, this carbon dioxide that is so essential for the process of photosynthesis. Okay. So what do I find in the middle? The mesophyll. I find palisade mesophyll. I find spongy mesophyll. And I find lots and lots of these air cavities. Okay, right. The slide over here. Okay, if we were to look at it, again, very often a question that I can ask you is, how are the palisade and spongy mesophyll adapted for photosynthesis? Okay, I'm going to just give you 1 minute. That's all we're going to have, 1 minute. And I want you to give me, all right, what I've just said. So you had to have listened. If you don't listen, oops, then you're nowhere. So what I want you to do, 1 minute. How is the palisade, how is it specially made for photosynthesis? And how is the spongy specially made for photosynthesis? 1 minute. You start now. How was that question? Were you able to think of what I said from the beginning? Okay. when we look at it, remember what I said, the leaf must be adapted to photosynthesis, which means its cells must have a specific structure to make photosynthesis work so much easier. Let's see what you had for palisade tissue. I was looking for the following. I wanted you to say, let's use pink, that they were elongated. Let's draw them. So we have palisade and we have spongy. I wanted you to say elongated, and your reasoning was a greater surface area. All right, which means that more light can trigger. All right, another thing I want you to say is right underneath the epidermis. That is an adaptation, because if it's right underneath the epidermis, light can come right through, okay? And again, we can get sunlight to get through to the epidermis as fast as we can. I wanted you to have said that it's filled with chloroplasts. Filled with chloroplasts, or many, let's rather say, many chloroplasts. Sounds a lot better when we say that. Many chloroplasts. Okay, and what are they? They're the organelle that has the chlorophyll. And what do we need for photosynthesis? We need chlorophyll. Okay, so when we look at the palisade, right, I hope you had those ideas. They are the ones that are more suited. Spongy mesophyll, they also have chloroplasts. Okay, they are also, one thing you could have put here, that they are thin-walled. What on earth does that do? All right, if it's a thin-walled, what can come in? Gases can come in so easily, carbon dioxide, and also water, which we need. Right, and what else does spongy mesophyll do? Hope you listen. Spongy mesophyll create spaces or chambers that are filled with air. Right, because what do we need these air chambers for? We need them, the carbon dioxide that's in the air, to be able to photosynthesize. I want to do one quick, the last layer, right? The last layer that we are going to find is going to be vascular tissue, right? Remember I said the veins. Now, if you have a look inside the leaf, what are we going to see? In the center of a leaf, we are going to find xylem and we are going to find phloem. Why? Because xylem needs to bring water to the top. All right, I'm going to actually use this diagram underneath. We can see it over here. What is the xylem going to do? It's going to bring water up from the roots into the leaf, because we need water for photosynthesis. So the water is going to go out, right? And underneath it, what are we going to find? Phloem. And what does phloem do? Phloem is going to take the food all the way down the plant so that the plant can also have a nice yummy meal. And there the phloem is going to take it down to the rest of the plant. Okay guys, it's time for a next quick break. Going to have a quick break, and then we get back. We are going to do some questions, all right, relating to the section of the leaf. See you back soon. Welcome back, grade 10s. Welcome to the last. We're going to do the last bit where we're going to look at a few questions. And I want to go straight into the questions so that it gives you a little bit of an idea of how we can ask you this section. Okay, don't be too scared when you see question 1. We'll go through it slowly with you. Okay, let's start. Question 1. The micrograph below, and all of you are probably going, what on earth is that picture that you have, ma'am? All right. The micrograph below shows a longitudinal section of a leaf. Guys, the diagrams that I gave you before, let me go back. Okay. Remember what I said to you? I said to you, this is a simple diagram that we use just to explain the leaf. The diagram now, this is what the leaf looks like when I actually take a part of the leaf and I look at it underneath the microscope. I'm going to give you a little bit of time. I'm going to give you all right, roughly 2 minutes. What I want you to do is to take that simple diagram, and I want you to try now and identify what parts of the leaf you can see. Okay, I'm going to give you 2 minutes because sometimes it is a little bit more difficult. Okay, guys, your time starts now. How did you guys manage? Were you able to look at a simple diagram and just take the concepts across to what an actual leaf is going to look at? Right, let's see. Let's start and see how well you guys did. When we take this diagram, I would suggest you start where we started in the beginning. We looked at the 3 different parts. We noticed that there is an upper part and there is a lower part. So you guys, very simple, you don't always have to use abaxial and adaxial, right? You could have used upper epidermis. Because what do we know? An epidermal cell surrounds it. What's the function of epidermis? To cover and to protect. And because we have a leaf, we were looking at a dorsiventral leaf. Dorsi means that there was an upper surface and there was a lower, all right, lower epidermal. Now, if I have a look, although I can't really see, if we try and look specifically, here there tends to be a bit of a space and here. But what would I call that space? That would be my stomata. Okay, now let's have a look at underneath the epidermis. We just said that this part over here is the middle. And that middle is called the mesophyll. Right, now the mesophyll. Now when I looked at the mesophyll, again, there were some that were elongated. You can see they're nice and long. And there's some that were round. The elongated ones we found at the top. And that was my palisade mesophyll. Okay. And the round ones was my spongy mesophyll. Right. So now, and what else did I notice? In the middle, I've got all these white spaces. Right. And what are those spaces? Chances are they are going to be air chambers. Because what did I say to you? What do we need a lot of in the leaf? We need to have a lot of air. So all these, any of these white ones here, all right, could be my air chambers. Okay, so far what have I got? Yes, I said my leaf was divided into one, the epidermises, the bottom and the top. So I've got my upper and my lower. Then I have the section in the middle where most of my photosynthesis is going to occur. And the cells that I saw there were my palisade and my spongy with a whole lot of air spaces. What's the third one that I'm looking for? The one that kind of like, if we look at the diagram here, kind of stands out. And this is this diagram in the middle over here. Now that diagram in the middle, when we labeled our leaf, that was your main vein, your midrib. Now if you have a look here, when we draw the leaf like this, okay, and we draw the main vein going there, and then we draw these little, all right, little midribs there, little veins that are going out on the side. Have a look here. There's a little one, and there's a little one. They're these ones that go out on the side. Now, let's have a look at the central one. What tissues did I have? I need to have two vascular types of tissue. The one tissue is xylem. Now, why do I know that this is xylem? Look at the size of the hollows. Xylem hollows, all right, always look a lot bigger than phloem. The reason being, phloem at the side, right, isn't as hollow as xylem, and very often, all right, it has this companion cell with it. Now, if you have a look at the red around here, if you have a look at the red, all right, all around here, that red, you'll see, is stained very much like that red. And that stain usually means that there is going to be lignin. Lignin means a strengthening tissue. But if you have a look, you will see there that it's only strengthened at the corners. So that is actually parenchyma, all right, and that parenchyma, right, forms, and I'm going to go back again. Let's go back to the next diagram. Can you see over here? The round the vascular tissue, we have a sheath, all right, that bundle sheath. And that bundle sheath is there to make sure that the vein and the phloem are protected and they don't collapse. And that is why it's made of parenchyma. And can you remember what parenchyma was? Parenchyma, C for corners, right? And what was that? We looked at our cell to strengthen in the corners, and that strengthening gave cell its shape. Okay, so if you look at it, in the beginning it's like, oh, what on earth is this? But if you slowly look at the sections, break it down section by section, as we did with our leaf, then it becomes a lot clearer to you. Okay, look at the next question. Right, our next question is coming up. Okay, guys, I'm going to now do a little bit of going from one slide to another because I'm going to show you the diagram, right? And then I'm going to quickly go to the answers, and I'm going to turn back to the diagram again, and then I'm going to go back to the answers again. Okay, the question is, study the diagram below and answer the questions that follow. Now, as soon as you see it, all right, things should stand up for you immediately. Okay, these are the questions that I want to know. How many stomata? That is the first giveaway that tells you what it is. How many of them are open? How many of them are likely to open at night? And then give me a reason for your answer. And then a skills one, what is the magnification for your diagram? Okay, I'm only going to give you a minute, 1 minute, and I'm going to hop to the diagram, hop to the question. Hop to the diagram, hop to the question. Okay, your time starts now. Hope you found that easy. It was a nice little easy one, but just reliant, most of it, reliant on what you saw. All right, so let's go through some of the answers and see how well you did. Okay, how many stomata are shown in this diagram? There are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Okay, so that question, just pure observation and knowing that stomata are made up of a guard cell, all right, that are specialized epidermal cells. Now the next question, how many stomata are open? Okay, so I use pink for the stomata, let's use blue. Okay, open. 1 is open, 2, 3, 4, 5, 6, 7. All right, so we got 7 that are open. Okay, so those 2 nice easy questions, all the answers there on your diagram. Okay, now comes the 2 little bit more difficult ones. Okay, question number C. How many are likely to be open at night? Give a reason for your answer. Guys, your answers should have been none. All right, and the reason why, what happens at night? Okay, the stomata close. And why do the stomata close at night? Right, because it's the whole osmosis thing that you were taught. What happens to the glucose? It gets converted as starch, all right, in the guard cell. And what happens to the water? It moves out at night. Water moves out. And when it moves out, then the guard cell closes. And I'm looking up and I'm seeing that we are out of time. I hope you enjoyed today's lesson and that you have a much greater understanding of plant tissues and where it fits in. Until next time, cherry bye.